Thanks, Jeyaprakash, for that kind introduction. I would also like to thank the organizers for this great opportunity to talk here in this session. I want to start my presentation with a disclaimer that this presentation is more of a user perspective of how cross-linking mass spectrometry can be combined with structural data obtained from traditional methods such as X-ray crystallography or clear-electron microscopy. So I'm not really going to the details of the technical details of cross-linking mass spectrometry. I'll give an overview of the workflow involved in cross-linking mass spectrometry, and during the later part of my presentation, I'll give two examples from our own work to demonstrate how we combine cross-linking mass spectrometry data with structural data from crystallography and biochemistry to address important biological questions. So, with that disclaimer, what is cross-linking mass spectrometry? So, it's a method by which two spatially proximal amino acids are covalently linked via a chemical cross-linker. And the identities of cross-linked amino acids are identified using high-resolution mass spectrometry. But why do we need cross-linking mass spectrometry? As you might agree, obtaining structural information for multi-subunit protein assemblies can be challenging, as often the intrinsic flexibility associated with the modular nature of protein subunits. And the cross-linking mass spectrometry is emerging as a powerful complementary tool to X-ray crystallography and clear-electron microscopy, mainly due to the fact that this can be applied to multi-subunit protein complexes of any size at any time. You do not require a lot of sample. It's rapid and economical, but you can generate a large amount of useful data. So what information the cross-linking mass spectrometry can provide? It can provide information on the tertiary structure of proteins. It can provide information on the protein-protein interaction interfaces. Depending on the type of chemical cross-linker one uses, one can obtain information either on the proximity of domains within a large multi-subunit protein assembly, or the identities of specific amino acids that mediate protein-protein interactions. It can also give valuable information on conformational dynamics. It can capture transient interactions that are functionally relevant. And the domain proximity information obtained from cross-linking mass spectrometry can also help in designing new constructs for further high-resolution structural studies with traditional methods. And this method is increasingly being used to decipher interaction networks in vitro and in vivo, and it routinely helps to assign proteins to densities in low-resolution structures. So what does it involve? First, the protein of complex, the protein of interest, or the protein assembly of interest, is cross-linked with a chemical cross-linker of one's choice. The colors here represent two different subunits, and once the complexes are cross-linked, then it's digested, often using trypsin, which will result in different types of peptides, linear peptides as well as cross-linked peptides. The peptide mix is then analyzed using liquid chromatography combined high-resolution mass spectrometry, otherwise known as LC-MS, to obtain high-resolution masses and fragment masses for the cross-linked peptide. The fragmentation spectra of all the peptides are then searched against a database to identify the cross-linked peptides. Once the cross-linked peptides are identified, the cross-links can be visualized using visualization programs such as the one developed in Yuri Rapsilber's lab, with whom we collaborate a lot, called XIView. You can also map the cross-links onto the available three-dimensional structures, or you can use the information obtained from cross-linking mass spectrometry as restraints for modeling. So as far as cross-linking is concerned, the protein cross-linking involves cross-links between a cross-linker and amino acid side chain. The reactivity of amino groups, thiols, and carboxylic acids of amino acids make these groups prime targets for cross-linking. What you see here is structure of a typical chemical cross-linker. And the crosslinkers have at least 2 reactive groups separated by a spacer. In this instance, the example that I have here is a crosslinker called BS2G. And this mainly reacts with primary amines, which means it can react with the primary amine of lysine or the amino terminus of a polypeptide chain. Upon reaction, the reactive group is then replaced by the primary amine of, in this particular case, the primary amine of lysine. This is just an example for a typical crosslinker, but we have a huge array of crosslinkers to choose from. The most commonly used crosslinkers are the amine-to-amine crosslinkers, such as BS3 and DSS, and carboxyl to amine crosslinker called EDC, and amine to non-selective photoactivatable crosslinkers such as SDA and sulfo-SDA. And these crosslinkers are classified based on what these react with. So that's why I was saying amine to amine, carboxyl to amine. So I'm going to introduce you to these 3 major classes of crosslinkers in the next few slides. The amine to amine crosslinkers are also called homobifunctional crosslinkers because the 2 reactive groups are identical and they react with same type of amino acids. And these amine to amine crosslinkers, there are different variants with different physicochemical properties, with different spacer arms, and some are water soluble, and some of the membrane permeable ones are not water soluble. And the amine to amine crosslinkers, in addition to reacting with amines, they also react with hydroxyl groups. So they can also crosslink serine, threonine, and tyrosine residue. BS3 is one of the most commonly used crosslinkers to study multi-subunit protein complexes, as lysine residues are commonly present on the surface, so it increases the chances of getting crosslinks. EDC is a heterobifunctional crosslinker, and it is a zero-length crosslinker. It covalently links the primary amine of lysine or the amino terminus of a polypeptide chain with the carboxylic group of either aspartic acid or glutamic acid. It's a very specific crosslinker, and it's ideally suited to map protein-protein interaction interfaces. So we can give information about the residues that directly mediate a protein-protein interaction. We use EDC quite a lot in our research. A sulfo-SDA is a photoactivatable crosslinker. It's a heterobifunctional crosslinker. It's a sulfo-NHS ester moiety, reacts with primary amines as well as hydroxyl groups, so it can also crosslink serine, tyrosine, and threonine, whereas the diazerine moiety can react not specifically with any amino acids. And there are also a few variants of SDA available with different spacer arms and also a cleavable disulfide spacer. So that's about the crosslinkers. And in a typical cross-linking experiment, one has to perform a titration experiment, varying the amount of crosslinker in the cross-linking reaction. And one might also need to change the amount of sample used in the cross-linking reaction. Oftentimes, one has to also vary the buffer conditions. Once the right condition where the cross-linking is efficient is identified, then you make replicates of this reaction and analyze them on the STS page and cut out the band that corresponds to the cross-linked species and do the digestion and go for mass spectrometric analysis. As I said earlier, I'm not going to go into the technical details of how cross-linked peptides are identified, but one point to keep in mind is that the peptide mix that you obtain after digestion is a very complex mix. So this includes ordinary peptides, which are unmodified, and just modified peptides where the crosslinker reacts only with one amino acid, cyclic peptide or internally bridged peptides, cross-linked peptides, and the combination of all the above. The cyclic peptides can give some structural information, mainly local structural information. For example, information about alpha helical region, whereas to gain insights into the domain proximity within a large multi-subunit assembly, or to get information about the actual protein-protein binding interface, we need to identify cross-linked peptides. So depending on the type of, depending on the sample that you use or the type of crosslink you use, one might need to consider enriching crosslink peptides using chromatographic methods. Once the crosslink peptides are identified, they can be visualized using visualization programs such as the one developed in Yuri Rapsenberg's lab called XIView. So this allows you to visualize the crosslinks in different ways, as a linear view or a circular view, or you can map the crosslinks onto the available three-dimensional structure. So that's kind of a very brief overview of the workflow that's involved in cross-linking mass spectrometry. Without introduction to cross-linking mass spectrometry, in the remaining time, I'm going to give you two examples to demonstrate how we combine cross-linking mass spectrometry data together with crystallographic data and biochemistry to address important biological questions. The first example that I have is the interaction mapping of the SCAR complex with its interaction with microtubules. The SCAR complex is a 3-subunit protein complex, and its subunits are called SCAR1, SCAR2, and SCAR3. It is an outer kinetochore microtubule binding protein, and it's one of the major regulators of chromosome segregation. As you might remember, to achieve chromosome segregation, the chromosomes need to be physically attached to the spindle microtubules. And the microtubules are dynamic structures. They undergo cycles of microtubule polymerization and depolymerization. During microtubule polymerization, the individual protofilaments, microtubule protofilaments, are kept in straight conformation. When the microtubule depolymerizes, the protofilaments peel off, taking a curved conformation. The mechanical strain that keeps the curved protofilament in a straight conformation is released in the form of force when the microtubule depolymerizes. To achieve chromosome segregation, this microtubule force needs to be harnessed to drive chromosome segregation along the depolymerizing microtubules. So the kinetochore needs to remain attached to dynamic microtubules as they assume different structures. The kinetochore microtubule attachment is mainly mediated by a protein complex called the NDC80 complex, shown here. Work from Andrea Musacchio and Ivan Nogales' lab showed that NDC80 complex binds microtubules by recognizing the alpha-beta tubulin dimeric interface. And this interface is sensitive to microtubule dynamics. When during depolymerization of microtubules, the individual protofilament peel off, taking a curved conformation, and at those instances, there will be a kink at the dimeric interface due to the curved nature of the protofilament. So that will influence how NDC80 binds microtubules. Various lines of evidence have showed that the attachment established by NDC80 complex need to be stabilized by the SCAR complex to achieve, to stabilize the kinetochore microtubule attachments and to achieve efficient chromosome segregation. So to understand how the SCAR complex binds microtubules, we determined the structures, crystal structures of SCAR core complex that was lacking the SCAR1 microtubule binding domain and the structure of SCAR1 microtubule binding domain on its own. Putting these informations together, it gave a kind of structural insights into the overall architecture of the SCAR complex. Alpha helical assemblies with microtubule binding domains flanking at the ends of the alpha helical assemblies. Microtubule binding proteins often bind microtubules via electrostatic interaction by recognizing acidic tail present in the tubulin. When we looked at the electrostatic surface potential of the SCAR1 microtubule binding domain, we saw possibly charged patches all over the surface. So this led us to think that the SCAR complex, instead of using a single high affinity binding interaction site for microtubule binding, it possibly uses multiple weak interaction sites to bind microtubules. Consistent with this hypothesis, when we mutated various lysine RNA clusters of SCAR1 microtubule binding domain, they showed reduced microtubule binding in vitro. So that kind of established that SCAR uses multiple sites for microtubule binding. But what regions of the alpha and beta tubulin or the microtubules are recognized by SCAR complex? To address this question, we collaborated with Yuri Rapsenberg's lab, and we carried out cross-linking experiments, two separate cross-linking experiments, where we cross-linked the entire SCAR complex with microtubules and then just SCAR1 microtubule binding domain with microtubules. And we analyzed the cross-linked product by mass spectrometry and identified the contacts between SCA1 microtubule binding domain or the SCA complex and alpha and beta tubulin. Here I've only showed the contacts, the cross-linked peptides that we identified between SCA1 microtubule binding domain and alpha and beta tubulin. I mean, it's been noted that the contacts that we saw between SCA1 microtubule binding domain and alpha and beta tubulin in the context of full-length SCA complex was almost identical, highlighting the specificity of the contacts that we see. And this clearly, this kind of confirmed that in fact SCA uses multiple contact sites to recognize alpha and beta tubulin. To understand the structural basis, we map these contacts onto the three-dimensional structures of alpha and beta tubulin dimer and on the SCA1 microtubule binding domain. So this analysis made us to make three main conclusions. First of all the sites on SCA1 microtubule binding domain that we identified to be critical for microtubule binding showed contacts with alpha and beta tubulin. Secondly, the data suggested that SCA1 might bind microtubules in multiple different modes. So there will be the binding modes could be different. For example, the clusters, the lysine clusters on SCA1 microtubule binding domain labeled as 1 and 2, each contact the glutamic acid clusters on beta tubulin. And these lysine clusters on SCA1 microtubule binding domain, if you look at the distance between the lysine clusters on SCA1 microtubule binding domain and the distance between the glutamic acid clusters on beta tubulin are near identical. So SCA1 can bind beta tubulin in two different ways. The 1 and 2 cluster facing 1 and 2 of beta tubulin, or if you just flip the SCA1 microtubule binding domain, the 2 and 1 of SCA1 microtubule binding domain still can make contacts with clusters 1 and 2 of beta tubulin. So it shows SCA complex can bind with microtubules in multiple different ways. And thirdly, most importantly, what we could show is that unlike the NDC-ED complex, the SCA complex contacts those regions of tubulin that are away from the dimeric interface, which is sensitive to microtubule dynamics. And these regions of tubulin are exposed to solvent irrespective of whether the protofilament is in a straight conformation or in a curved conformation. So SCA can bind microtubules, both polymerizing and depolymerizing microtubules in a conformation independent manner. So putting these information together, we could suggest that SCA, with its multiple microtubule interaction sites that can engage with microtubules in multiple modes, can remain attached to microtubules as microtubules assume different structures. And it is the SCA complex that confers its ability to remain attached to microtubules to the kinetochore and hence to achieve accurate, efficient chromosome segregation. So the second example that I have is the interaction mapping of the chromosomal passenger complex that consists of borealine, cervigin, insen, and RB kinase and its interaction with nucleosome. The chromosomal passenger complex, which is commonly known as CPC, is one of the major regulators of chromosome microtubule attachments. The chromosome microtubule attachment is a stochastic process. As a consequence, it's prone to incorrect attachments. And these incorrect attachments need to be eliminated before the chromosome cystochromatins start to segregate. And the chromosomal passenger complex, CPC, eliminates this incorrect attachment by phosphorylating the microtubule binding components of the kinetochore, such as the NDC-ED complex and the SCA complex that I just introduced you in the previous example. So as I had mentioned earlier, these microtubule binding proteins bind microtubules by recognizing via electrostatic interaction, and CPC eliminates incorrect attachments by introducing a phosphorylation in the microtubule binding interfaces of NDC-ED and SCA complexes. So the phosphorylation will change the electrostatics and hence will weaken the interaction between NDC-ED and SCA with microtubules. So to do its function, the CPC needs to associate at the centromere region of the chromosomes. Several studies have shown that two histone modifications, just don't worry about the fine details of this diagram, all that I want to show here is that two histone modifications have been shown to be crucial for recruiting CPC to the inner centromeric region. And particularly, one of the histone modification was recognized by the surviving subunit of the CpC. Okay. However, removing the histone modifications was not sufficient to remove the CpC completely from chromosomes. It only dislodged from the centromere, but the CpC is still associated with chromosomes. So this made us to ask a question, then how does CpC associate with chromosomes? By doing extensive biochemistry and biophysical characterization, we could show that CpC can bind nucleosomes, bind unmodified nucleosomes in a phosphorylation independent manner. So CpC has intrinsic ability to bind nucleosomes. Using surface plasmon resonance experiments, we measured the binding affinities of CpC for unmodified nucleosome and phosphorylated nucleosome, and it showed that CpC binds unmodified nucleosomes with about 380 nanomolar affinity, with phosphorylated nucleosome with about 90 nanomolar affinity. So to understand how CpC binds nucleosomes, we reconstituted the CpC nucleosome complex in sex exclusion chromatography, both with unmodified nucleosome and nucleosome containing histone S3 threonine 3 modification, the phosphorylation. And then we performed cross-linking and analyzed the cross-linked sample. And just for clarity, I've shown here only the interaction between survivin and borealine subunits and the histone subunits of the nucleosome. Consistent with in vitro data, what we could see is that borealine makes extensive contacts with all the 4 histone subunits. Particularly strikingly, it was also making contacts with these highlighted regions of histone H2A and H2B, which contributes to an acidic patch on the nucleosome, which is commonly involving protein-protein interaction. I'm going to show that acidic patch in the next slide. Whereas surviving histone interactions are confined mostly to the N-terminal tail of histone H3, and the histone phosphorylation happens in the N-terminal tail. And this kind of confirmed our previous studies that in fact survivin can directly bind the modified histone H3 tail. Okay? But then this was completely a new information that borealine makes extensive interaction with the nucleosome. So if you map these contacts onto the three-dimensional structures of nucleosome and the CpC, this is what we see. Here, the nucleosome and the CpC structures are shown in surface representation and color based on the electrostatic surface potential. What we see is that the positively charged N-terminal region of borealine contact this acidic patch contributed by histone H2A and H2B, and this site is commonly involved in mediating protein-protein interaction. And the borealine loop region that connects the N and C terminal region of borealine makes extensive contacts along the DNA histone interface, highlighted here as dotted circles. Okay, and the docking of N-terminal region of borealine into the acidic patch will bring the surviving DAR domain close to the histone H3 tail, and it will facilitate the binding of survivin and the histone H3 tail. So
2 more minutes, JP.
Yeah, so this can establish that CpC can bind nucleosomes in a phosphorylation independent manner exclusively via borealine. So this made us to kind of propose a model that during early mitosis, when there is no histone modification, CpC associated with chromosome in a phosphorylation independent manner. As the cells progress into metaphase, the histone modifications end CpC to the centromere. So what I've shown just now are just 2 examples to demonstrate how we combine cross-linking mass spec data with structural data from crystallography and biochemistry. But the scope or applicability of the cross-linking mass spectrometry extends beyond what I've just discussed now. So you can get information about protein complex topology of a huge multi-subunit protein assembly. You can use cross-linking mass spectrometry data for de novo modeling, to quantify conformational dynamics, and to look at protein topologies inside cells. So I would like to remind you that there's going to be a talk from Francis, who works with Yuri Rapsilberg, He'll be giving a talk tomorrow about cross-linking methods to look at proteomics in cells. So with that, I would like to thank our collaborators, Yuri, Rapsilver, and Juan. They are extremely fantastic collaborators. It's a privilege to have them next door, actually. By the way, Yuri also has a lab in Berlin. In fact, that's his main lab. But it's such a privilege to have them next door, and it's always a fun collaborating with Yuri and Juan. And all the work that I discussed was mainly led by Alba and Bethan, extremely talented postdoctoral researchers in my lab. And I'll thank you for your attention. I'll be happy to take any questions.